Okay, what I'm going to talk to you about today is basically how I spent my summer vacation. 2 research cruises and then another research cruise for a class that we're teaching. The first research cruise I was on was for a program called Census of Marine Life. This is a 10-year project that started in 2000. It involves 80 nations and 2,000 scientists. The purpose behind this is to assess and explain the diversity, distribution, and abundance of marine life. There are a number of reasons for doing this, but thus far, in the 10 years of research, and not all the data has been analyzed yet, 5,600 new species in the oceans have been discovered. They're also rediscovering old species that they thought were extinct. For example, a shrimp was found swimming off of Australia that had been thought to be extinct for 50 million years. And information from this project is critically essential for making a database against which to assess the impacts of global warming. You have to know how things are before you can determine how things have changed. So the purpose of this Census of Marine Life is to determine how many species there are in the ocean, how many of each species, and of course it's just a selection of those, and where they're found. So there are 14 different projects associated with the Census of Marine Life, and the one that this cruise was associated with is called MAR-ECO. MAR stands for Mid-Atlantic Ridge. And this cruise was to investigate the diversity of life along the Mid-Atlantic Ridge. This research involves 16 nations, and the project leader of this is Odd Axel from Norway. The Mid-Atlantic Ridge, this is the longest mountain range on Earth. It extends from the north all the way down to the south, so it basically crosses across the oceans. It's 75,000 kilometers long, and in some locations, it's 2.5 kilometers high. The reason it requires 16 nations is that they've kind of divvied up the Mid-Atlantic Ridge. Various nations are studying various portions of it. Our particular cruise was up here in the northeastern portion of it. The cruise was on the Fisheries Science Vessel Bigelow, which is a 63.6 meter ship. The chief scientist on this cruise was Mike Vecchione. He's with the National Marine Fisheries Service and the Northeast Fisheries Science Center. The co-chief scientist was John Galbraith, so he did all the net trawling, and that was really an amazing amount of work that he had to do. So each nation involved in MAR-ECO gets funding from their nation, and this particular cruise was funded by the National Marine Fisheries Service and NOAA's Ocean Exploration Program. This is the cruise track of the Bigelow. We started out in Gloucester. We're going to our trawl site, but had to divert to Newfoundland. We lost all internet communications. Because this portion of the Atlantic tends to have a lot of storms, we desperately needed the internet so that the ship's crew could track the storms and get it out of there if we had to. In addition, the scientists really needed the internet as well, particularly for me because I was finishing up a grant proposal while I was out there. So where we were specifically was along the Mid-Atlantic Ridge, but we were along a fracture zone that actually runs parallel to the ridge. And this is called the Charlie Gibbs Fracture Zone. So the idea behind this cruise is to study the biodiversity of life on north of the ridge, where there's northern, highly productive cold water, and south of the ridge, which is more southern, not as productive, warmer water. This being my first cruise up in the North Atlantic, I also saw my first icebergs, which was very exciting. They let us get close to the first 2 icebergs we saw, and then like any normal research vessel, they did their very best to avoid all future icebergs. So these are my only 2 pictures of icebergs. The trawl nets that we used, we were doing both pelagic trawling in the midwater, and benthic trawling on the bottom. And the trawl nets were stored on these 2 spools. What you see here now is the pelagic trawl. The pelagic trawl has a sled on it, and at the end of the sled are these 5 separate nets. Each of these nets has a cod end or collecting vessel on the end, and they could be triggered individually, meaning that on a single midwater trawl, we could entrawl the entire water column, usually from 1,000 meters depth down to almost the bottom of the mid-Atlantic ridge in this region, which was about 3,500 meters. So when the trawl nets were brought to the surface, they were sorted into these highly advanced trawl buckets. Each trawl bucket was carefully labeled because it was absolutely critical that these trawls were maintained separately, and we knew which trawl each sample came from. So this is sorting in the lab, and what I want to show you here is that this is a daytime midwater trawl at the shallowest portion of the midwater trawl. Because of vertical migration, a lot of times during the day, those shallow trawls came up fairly empty. This shows the emptiest trawl that we got at night. Now, the way the cruises worked is that we were put on 2 watches. One group worked from noon to midnight, and the other group worked from midnight to noon. And guess which watch I was on? I was on the midnight to noon. So we had to spend a huge amount of time sorting samples. These are a sample, this is all the same species. This is a little fish called Cyclothone that Tracy Sutton sampled, and he's got a rather bizarre sense of humor. So when all these samples have to be sorted, then they have to be weighed and stored in various storage media, either formaldehyde or ethanol or frozen for DNA samples. What Tracy wrote here on his little sample note is, please measure each one. There are 769 fish on here. This is, of course, the night crew who has to do this. But luckily, it also says over, and on the back it says, just kidding. So this is what one of the nighttime trawls looked like. Each and every animal had to be sorted out of here. The reason I was asked to go on the cruise is that I have expertise with identifying shrimp. So I was asked to come out and sort out the shrimp, key them out as far as I could. So eventually, the shrimp were sorted out into their individual groupings, and most of them were red, so not that easy to identify. And the same thing was done with the fish. And then, as I said, the samples were stored for a variety of reasons, to look at stomach contents for the fish, to store in ethanol, to store in as a frozen sample for DNA samples. Now, as you might notice in a lot of these pictures, working in the lab, a lot of people are very warmly dressed and wearing foul weather gear. As well, anybody who's working on this side was wearing their boots. The reason is this is a fisheries vessel. So normally on fisheries cruises, the fish are brought in on a conveyor belt, they're measured, weighed, and then flung out. So here was an open trough going back to the ocean. In addition, the smaller ones were simply flung on the floor because on this side of the wall, there was a 1 to 2 foot gap between the wall and the floor at various spaces. So that when you flung the fish on the floor, eventually it would also go back out to the ocean. But what this meant is that when you're working in this lab, if it was 47 degrees outside, it was 47 degrees inside. And if you're working on this side, in rough seas, which they always have in the Northeast Atlantic, water was constantly sloshing up over the floor here. So you had to wear your boots so that your feet wouldn't get soaked with this freezing water. But luckily for me, I suffered in silence, as is my custom. So I have been midwater trawling for 25 years, and really, I didn't agree to go on this cruise because of the midwater trawls. There's not that much I haven't seen. There are some interesting anglers that sometimes come up, but for the most part, I was really looking forward to the benthic trawls. But this came up in one of the midwater trawls. Now, this obviously didn't fit in one of those little cod ends. And we knew that sometimes we would catch big stuff that wouldn't go in the little cod ends. So in addition to the people who were sorting in the lab, there were also a couple people whose responsibility it was to go through the 5 nets and see what they found. This is what they found in one of them. This is a dealfish. This came from one of the shallower sites. The depth at the site that this fish was found at was about 725 meters. And they think this fish was caught at about 400 meters. So it's not that an unusual fish, but I've never seen anything like it before. In addition, it has a very protrusible snout. So the snout extends out. And what really interested me the most was this enormous eye, because I work on vision. But unfortunately on this cruise, the entire cruise was to count and sort animals. No other physiological studies possible. And like I've said over and over again, it's really hard to get any kind of information from a dead eye. Some new species were also discovered on this cruise. This is an isopod. It's been sent out for taxonomic analysis, but they're pretty sure it was a new species. And we didn't really have any worm experts on the cruise, so they thought this might be a new species, but again, it has to be sent out for taxonomic analysis. On the cruise, we had David Shale, who did some of the filming for the BBC Blue Planet series, and so he took this spectacular picture of this worm, which has one of the most bizarre snouts that I've ever seen. So eventually word will come back on whether or not this unusual beastie is also a new species. So as I said, I was mostly out there for the benthic trawling. This is the cod end of the benthic trawl. It's basically just a big sack. It has to be fairly carefully rolled up to go inside the net when the net is launched, so that when it's released, it deploys properly and opens up. These nets are extremely complex, which means that no scientist, except for the chief scientist, was still out on deck while they were being deployed. A very smart move on their part, I think. And so the net is deployed over the side. It has this fringe on it there to help the cod end. That increases the resistance, the surface area, so that helps keep the cod end up off the bottom a little bit. In addition, it has these floats on it. Now these cod ends are stored on those spools that I show you. So with every launch and recovery, the floats have to be put on and then removed. These rollers serve to roll along the bottom and kind of churn up the bottom a little bit. So as the net goes through the water, the bottom is churned up and stuff is then basically turbulent flow into the net. So as I said, this is a fairly complex net, and you see it has all these different cables associated with it. And what you're going to see in a minute is two of the crew are going to attempt to untangle or unravel these cables. It's actually a fairly dangerous job because there's a huge amount of tension. Now this is what I wanted to show you. There's no way that spool holds 10,000 meters of cable. So the reason they're unraveling this cable like this is so that they can separate a two-cable system here. So now you can see that there's actually a separate cable attached here. Once they have them untangled and untwirled, the net is going to be let out, and then a crewman from here is going to grab that cable, detach it from the spool, and attach it to another cable that's attached to a huge winch below the deck of the ship that does hold 10,000 meters of wire. So with this two-cable system, that ensures that the net stays open, and that also means that there's enough cable available to send the net down to the depths that it needs to be sent to. Now, this being a Census of Marine Life project, every animal that comes up in the net has to be counted. With the pelagic trawls, not that much hung up in the net. In this benthic net, as it was going down, it did catch animals. So when the net was put back on the surface, they would bring in a little bit, and then all of us had to go through the net, sort out the animals. They would bring up a little more, and eventually we would get to the cod end. So this is the cod end of the net, and it's full of our samples. Oftentimes the samples look like this. A mixture of, this is actually quite a large jellyfish here, mixtures of echinoderms, shrimp, and fish. Like I said, every one of these had to be sampled out. I was in charge of the shrimp. We had somebody in charge of the jellies, somebody in charge of the fish. So most of the benthic trawling, most of the benthic sampling involved doing the samples outside on the freezing cold deck, but as usual, I suffered in silence. So in sampling through these nets, there were actually some very, very interesting items. And like I said, this is why I came on this cruise. I have done almost exclusively midwater trawling. I do very little benthic trawling, and I rarely have been on a cruise where there's really deep benthic trawling going on. So you can see them removing a fish here from the side, and also notice this bizarre creature. This is a slickhead, not him, but this fish here. This is John Galbraith, who's holding the slickhead. This is the biggest slickhead he's ever seen. Normally, he sees them and they're about 6 to 7 inches long. So he was really thrilled to get one of this size. This is a sea urchin. It's actually a very soft-bodied urchin. So when it comes to the surface, eventually all the water leaks out of it and it sort of becomes like a little sea pancake. In one trawl, it was packed with these football-shaped things. These are echinoderms, meaning that they, even though they don't look like it, they're related to sea stars and sea urchins. They look more like slugs, more like anything else, but this being Census of Marine Life, these had to be removed from the net and counted as well. But as we quickly discovered with sea urchins, when you have a basket of sea cucumbers, I'm sorry, these aren't urchins, these are sea cucumbers, you eventually end up with a basket of sea cucumber goo. So we realized that we had to individually count these as they were going into the basket because we didn't have enough graduate students on board to volunteer to separate the sea cucumber goo. And so once we counted them on the surface, once we counted them outside, then we could simply go and measure these and get the biomass. So sea cucumber goo, almost as disgusting as disgusting hagfish goo, but not quite. So some of the interesting fish that came up in this trawl, this is a chimera. And a chimera, it's in the same classification as a shark, but it's called a chimera because it shares both shark-like and bony fish-like characteristics. So like the shark, it has a cartilaginous skeleton, but like a fish, it has an operculum over its gill vent. This is the long-nosed chimera, and it has a very, very distinct lateral line that goes over its entire body. Lateral lines are motion detectors, so this is a very sensitive motion detector. It also has these very unusual wing-like fins and a very, very nasty spine with a venomous tip. So most of these trawls, you had to be very careful, carefully sort out, kind of spread things aside to see what was in the trawl before you actually grab something. This is another chimera. It's called Hydrolagus. It is the biggest one of this species that's ever been seen. As you can see, it has a vicious spine right here. And this is being held up by Dr. Tracy Sutton. Tracy used to be a fish biologist here at Harbor Branch. He's now at the Virginia Institute of Marine Science, and he's the one who invited me to participate on this cruise. This is a lizard fish, and this fish is a vicious, vicious carnivore. It's called Bathysaurus, so it's almost like a living fossil. It has one of those old dinosaur-y type names. It is the world's deepest living superpredator, meaning that it eats any fish it sees, including its own species. It's also hermaphrodite, and what I find very interesting is its extremely strong eye glow, even when it's dead. So that means these eyes, like a lot of animals adapted for life in the deep sea, has a reflecting mirror behind the eye, and that basically doubles or triples the sensitivity of the eye. Mike Vecchione is one of the world's experts on cephalopods, which include octopus and mollusks. And in one of the benthic trawls, he got this 5-foot octopus called Dumbo. This is a little caption that he wrote for a website that this picture went on. And it says, Mike Vecchione retrieves the big Dumbo, a Cirrothamomagma, approximately 5 feet long from the catch. The Dumbo is not the one wearing a helmet and life vest. So remarkably, it was intact enough so that Mike could actually identify to genus and species. What you see here on the left, that's David Shale. So he's the Blue Planet photographer who was out on this cruise to take images of these animals. There is a very important reason for taking these images, not only because other people like to see them, but also for taxonomic identification purposes. It really helps if you know the original color of the animal, if you know what it looked like before it crumpled and shrank when it was stuck in formaldehyde or ethanol. You also had to be careful in the trawls in looking for some of my beloved crustaceans. This is a Neolithotid crab. These big ones weren't actually that dangerous because they are quite big and easy to see, but we got a fair number of very small Neolithotids, which you often didn't see until they poked into your finger. So this is why I came out on this cruise, to see these big, beautiful crustaceans. These crustaceans are bathypelagic. That means they live in the water column, but very, very close to the bottom. Being so close to the bottom, we've seen these in shallower waters on our submersible dives, and we know that sometimes they actually rest on the bottom. So that's one reason why they can afford to have such a large size, is that they don't have to continuously spend energy to maintain their position in the water column. The other reason they have this large size is very likely due to the lack of predation down there. There's not as many predators down on the deep sea floor, so they live longer, and the longer they live, the bigger they get. So in these benthic trawls, we got a fair number of these big red shrimp, as well as the more standard-looking animals that you expect to find on the bottom, a variety of lobsters and other really armored shrimp. And it's this lobster in particular that I found extremely interesting because I've never seen one before. I've only heard of them. This is a blind lobster. It doesn't have any eyes. So somewhere along evolutionary history, it completely lost its eyes. This was also considered a relatively rare lobster, but on this cruise, we got a fair number of them in most of the benthic trawls. We also got a fair number of spider crabs. These are sea spiders, and they're in the family Pycnogonidae. I often get them in the midwater in my trawl nets, but they're about a ½ inch long, and a lot of them are even microscopic. These are enormous, and who knows what they do when they're actually crawling along the bottom. Now, because of bad weather, we only got 5 benthic trawls, and 2 of those hung up on the bottom. So the work that John Galbraith had to do to trawl this net along with the NOAA crew was just phenomenal. We were at the Mid-Atlantic Ridge, so it's a mountain range. It's an underwater mountain range. And he was trawling along this underwater mountain range, moving that net up and down as we were trawling. So it's really quite amazing that we didn't hang up on the bottom more often. Now, in addition to the spectacular animals that we got in our trawl nets, we also saw some charismatic megafauna, which is whales and dolphins. And I'm not showing you this just to show you yet another picture of a whale, but what this whale was doing was showing some behavior that I've never seen before, and scientists on the cruise had never seen before. I'm sure our whale and dolphin research group has seen this, but we haven't. And what was going on here was a mating display. So this is a male, and he was slamming his fin. That's me falling. Now I'm back up again. He was slamming his fin back and forth on the water, and this went on for about an hour. Now he's doing the 2-fin salute. We are about a quarter mile away from here, so I'm actually at the limit of my camera's ability to take pictures. Now it's switched from the fin display to the tail display. So that went on for about another half hour, and he got further and further away from the ship, and eventually, too far away for us to film, then a female came by. We're assuming it's a female. It could have been a male, but... And the whale did breach, and Mike Vecchione got the spectacular shot of it. I, unfortunately, was switching from my video camera to my still camera just as the whale breached, but Mike was very generous with his photos. So that was the Census of Marine Life cruise, and don't have much to report on the data yet. We're still in the process of analyzing up, and I'm due to go up to Virginia probably in the middle of April to finish analyzing the midwater and the benthic samples. So stay tuned next year for the results of the Census of Marine Life cruise. So I left the frozen tundra of Greenland and spent a week on shore, and then did my next cruise off of the Bahamas. And the basis of this cruise was a result of some research that we had done on an earlier cruise. The reason we were able to do this research is because of the ability of the Johnson Sea Link submersible to collect animals, and in addition it has this wonderful sphere at the front which we could use to help in our collections. What we were able to do is put red lights on the Johnson Sea Link, and then I had deployed some traps. We were able to go down under red light and collect those traps filled with crustaceans. In addition, the pilots were so good, they could also collect some crustaceans in a claw. And you might say, well, what's the big deal? Lobster fishermen, shrimp fishermen collect these animals all the time. Well, the problem is when you bring them to the surface in light, you completely blind them. These are animals designed for vision in an environment where there isn't very much light. They have extraordinarily sensitive eyes. You bring them to the surface and expose them to the surface light levels or ship lights, it's like us staring at the Florida sun for 5 minutes without blinking. Do irreparable damage to our eyes. With the Johnson Sea Link, we could collect them in the dark, bring them up in a temper-insulated container, and for the first time ever, determine what the visophysiology of some of these deep-sea crabs are. Remarkably enough, we found one species, a squat lobster, that has both a blue-sensitive and an ultraviolet-sensitive visual pigment. Now, most of the crustaceans that we worked on had a single blue visual pigment. This is what you would expect for a deep-sea animal. As light goes in the ocean, the long wavelengths are absorbed and the short wavelengths are scattered. What remains is blue light. So when you're in a light-limited environment, it doesn't do you any good to have colored visual pigments because the only light that's there is blue. So get rid of all visual pigments and just have a lot of blue visual pigments to make yourself as sensitive as possible to the available light. So this is actually what I was expecting. I was expecting highly sensitive animals, and I was expecting a single visual pigment. I was not expecting a dual visual pigment system. However, I had found in some species of deep-sea pelagic crustaceans, those are crustaceans that live in the water column, 6 out of the hundreds of species that have been studied have 2 visual pigments. And that turned out to be related to their bioluminescence. Well, Gastropticus, the animal with the 2 visual pigments, is not luminescent. However, we always see it associated with these corals or colonial anemones. So one of the questions was, maybe these corals or anemones put out short wavelength luminescence that would help the crab find its preferred habitat. Another question that arose from this cruise had to do with some information we got from an isopod. An isopod is like a deep-sea pill bug. They also have fairly large sensitive eyes. What's unusual about these eyes is that they are phenomenally slow. And by that I mean it's like a camera with a very, very long shutter speed. The way we measure a shutter speed in a biological eye is to measure something called flicker fusion frequency. In our eyes, our flicker fusion frequency is 60 cycles per second. That means that something flashing at 60 cycles per second or slower looks like individual flashes. something flashing at 70 or 80 cycles per second looks like a steady glow. That's why fluorescent lights don't drive us insane, because they're flashing at 200 cycles per second. So we can't see those flashes until the light starts to go bad. Then its flicker gets much slower, and eventually it's that slow, annoying flicker that makes you find it necessary to change your light. These animals have a flicker fusion frequency of 3 cycles per second. So what this means functionally is as follows. For our eyes like ours, with the high flicker fusion frequency, this is what we would see at a nighttime scene. We'd see these little pinpoints of light. For an animal like the isopod, with the extremely high flicker fusion frequency, this is what it would see at the same. All this contrast is here in this picture. We just can't see it because our eyes aren't sensitive enough. They, with their extremely sensitive eyes, can see that contrast. And that's really important when you're a deep sea animal living where there's not much light. The problem is, if this were a luminescent animal and it moved from here to here, we could tell that it was going from here to here. And an eye that's this slow, it's like a camera with the shutter held open. What they see is a streak. So they don't know whether the animal is moving from here to here or from here to here. So what this means for this isopod is that they have an eye that is so slow, the integration time is so long, that it's unlikely they can track a moving object. However, isopods are known to be scavengers and detritivores. That means they eat dead stuff that falls to the ocean bottom. And one of the theories has always been that when decaying matter falls to the bottom, it's colonized by bacteria. A lot of marine bacteria is bioluminescent. So some scientists have wondered if there might be mats of decaying matter on the ocean bottom that's actually dimly glowing, that we've never seen because our eyes aren't sensitive enough. Both of these relate to bioluminescence. But unfortunately, very little is known about deep sea benthic bioluminescence. We know very little about bioluminescence of animals that live on the bottom. We know a lot about bioluminescence for animals that live in the water column because we have special nets designed to bring them up alive. But we can't do the same thing with bottom-dwelling animals. The benthic trawls and dredges really do a lot of damage to them, doesn't bring them up alive, and it's very hard to measure bioluminescence in a dead animal. In addition, where we did our work with these crustaceans, there's a lot of bottom habitat. It's hard bottom, there's a lot of coral standing around there. A benthic trawl and a dredge would do irreparable damage to this habitat. It would take it hundreds of years to recover. So trawling and dredging wasn't even an option. And most of the studies have been done with submersibles and ROVs using light, and that light would swamp any bioluminescence. So there really was very, very little known about deep sea benthic bioluminescence. So my colleagues and I got together and wrote a grant to the NOAA Ocean Exploration Program to study bioluminescence on the deep sea floor. And we were able to do this and get funding for this because we had access to the Johnson Sea-Link submersible. In addition to using the cameras, both a video camera and a still camera on the outside of the submersible, the scientists sitting inside of the submersible could also use low-light video cameras and low-light color cameras to attempt to get in situ, meaning in-place pictures of bioluminescence. In addition, I could continue my vision studies because we could still use these red lights to bring up animals that were caught in my traps. We can bring these animals up alive because of this big bio box that's on the front of the JSL. It's made of very thick plexiglass, meaning that it's temperature insulated. So if we were down there and stimulated something with the claw and it luminesced, we could pick it up, we, this being the royal we, of course, with the sub-pilots doing all the work, bring it up, put it in the bio box. The bio box would seal, bring them up in cold water, they would come up alive, and we could study them in the lab. So I was the chief scientist on this cruise, and my collaborators were Steve Haddock from Monterey Bay Aquarium Research Institute, Sanka Johnson from Duke University, Chuck Messing from Nova Southeastern, right here about an hour south, and Edie Witter from Orca. And this was a NOAA signature expedition, so there was a web page associated with the cruise that has a lot of pictures, as well as an @sea web page on FAU's website as well. In addition to my colleagues, Gabby Barwright, my graduate student, came on this cruise, and this is a picture of her making her first submersible dive. And we always bring an educator at sea, and this time it was Angie Lewis, who's a science teacher with the local Marine Oceanographic Academy, doing her first submersible dive. And as volunteers, they spoiled me for any future volunteers because they were so good. So what we were able to do is Sanka Johnson researched and found that there was this new Nikon D3 camera available that could be pushed to film speeds of 25,000. Now, most of the cameras that you and I use have ASAs of 200 to 400. So this meant this color camera had remarkable sensitivity. In addition, we used it with a lens that could be opened up to a very large aperture. So by using this from inside the Johnson Sea-Link submersible, as well as in the lab, we had the opportunity for the first time to take color pictures of the animal while it was luminescing. Now, while it's nice to use the eye and say, well, this looks kind of bluish and this looks kind of greenish, being scientists, we also needed a more quantifiable method of looking at the luminescence. And so the luminescence was recorded with this ocean optic spectrometer that sort of verified what our eyes were telling us. So this research was done on Little Bahamas Bank and Northwest Providence Channel. We chose these two sites for two reasons. Number one, the Little Bahamas Bank, there are lithoherms there, which are mounds, and we talked to John Reed and Chuck Messing about a good site where there was a lot of benthic habitat, a lot of animals living on the bottom, because this was going to be an overview of bioluminescence in the deep sea benthos. So the more species we could test, the better. And Chuck and John had both been to this site before, knew there were a lot of benthic animals living there. So that was a great site for the overview studies. However, there's a lot of current action at this site, meaning that if any food was falling to the bottom, it was likely to get swept away and we wouldn't get any mats. So we also chose a Northwest Providence Channel site, very, very different habitat, very fewer currents. And as anybody who goes to sea will tell you, once you write a grant to study something, it often disappears. We wanted to study the isopods at this site, and we saw not a single one. This is the first cruise we've ever been on where we didn't see any isopods, and it's very likely because we had them in our grant proposal. We've seen isopods at all the other locations that we've been to. So on this cruise, we could not study decaying benthic mats. There weren't in this region, and coincidentally, there weren't any isopods either. So that question remains to be answered. But the question about an overview of bioluminescence was answered on this cruise. We managed to study a lot of different animals. We discovered a new species, and it's again being the royal we, because it was actually Chuck Messing who discovered this. He discovered a new species of crinoid. And a crinoid is a sea lily. It's in the same family as sea stars and sea urchins. It's attached to a stalk, and it has this beautiful lily edge on it. See if I can get the lights down a bit. So Chuck is a crinoid expert. He's also an expert on a lot of the benthic organisms, and so he was able to tell us as we were collecting things with the sub what a lot of them were if they weren't crustaceans or fish, which was the only thing that most of us knew. In addition, we found another species of crab that had an ultraviolet photoreceptor. So this is Humanita picta. It's found all over the Gulf of Mexico, along the east coast of Florida, off the Bahamas, and it also has a blue sensitive and an ultraviolet sensitive visual pigment. So now we have two crabs in the same family. Both have a blue and a UV sensitive visual pigment. And the question is is there ultraviolet bioluminescence in the deep sea benthos? The answer appears to be no. We measured 100 different species. This is one of the dominant species at the lithoherm site. It's actually a colonial anemone. This colonial anemone, you can see the little buds here. These are little tiny anemones. It actually forms a skeleton matrix like a coral. So its common name is the golden coral, although it's not a coral, it is an anemone. It's called Gerardia. It forms these big tree-like structures, and it has spectacular luminescence. Kind of this blue-green color here. This was filmed by Sanka Johnson in the lab. So here is the animal in red. So this was filmed entirely by the light of the animal with this new Nikon low-light sensitive camera. We found 2 species of solitary anemone, meaning not colonial like Gerardia. It's called the Venus flytrap anemone because it does somewhat resemble a Venus flytrap, and it puts out a luminescent slime. So this anemone grew on other hard structures at the lithoherm site. It grew on corals. This is actually one of those dead zoanthid trees. There was another species of solitary anemone that we always found on the back of a hermit crab. It's a different species. It hasn't been identified yet. It's been sent out for taxonomic analysis. But it also puts out this beautiful blue-green slime, just like the other species. All 5 species of bamboo coral that we tested were luminescent. But interestingly enough, no non-bamboo corals were luminescent. So the black corals were not luminescent. Some of the red corals were not luminescent. Only the bamboo coral appears to be luminescent. And again, it's a spectacular luminescence that goes up and down the colony and is also kind of blue-green in color. We found 3 species of sea pen, and these actually have a beautiful, almost emerald-colored green luminescence. So these are the farthest green-shifted luminescence of any of the species that we studied, a distinct emerald green color. And Sanka was also able to get pictures of this crustacean putting out its bioluminescent spew. Luminescence in these crustaceans is not new. It's been known for a while, but it's never been filmed before with the image of the animal. And I was studying the vision of these animals. They came up in my traps, Parapandulus, Heterocarpus, and Plesionica. When I was through with them, I gave them to Sanka to see if he could take some pictures of the luminescence. So all the blue light here is the luminescence from these shrimp. It's spectacularly bright. When I get them species that give off luminescent spew in my trawl buckets, it's actually bright enough to read by. What they use this for is a predator defense. It's like the black inky spew that a squid puts out in a tide pool. But when you're in the deep, dark ocean, putting out a black spew isn't really going to do you any good. But if you can spew this incredibly bright fluid right in the face of your predator is going to momentarily stop while you can go tail flipping away. In addition, in some species, the spew is actually a little bit sticky. So it actually sticks to the predator, attracting predators to the predator while the prey gets away. But what we discovered on this cruise is that bioluminescence in deep-sea bottom-dwelling animals, deep-sea benthic animals, is actually quite rare. We tested 100 species and found that only 14 of them were luminescent. Now, this was unexpected because in the literature you can find a lot of papers on luminescence of animals that live in the water column. For animals that live in the water column, about 90% of them are luminescent. Compare this with 10% of the animals in the deep-sea benthos. So deep-sea bioluminescence is not that common unless you have a lot of the species that happen to be luminescent. However, because we were doing our studies from the Johnson sea link with that spectacular spear on the front, Sanka Johnson made a very interesting observation. He was sitting down there and setting up the Nikon camera in the dark for a long integration time picture. And as he was sitting there, he noticed these blue sparks on the green luminescence from the Gerardia. And what he noticed is that it appeared that luminescent organisms, small organisms in the water column, were constantly striking these benthic structures, these big benthic trees that were standing up in the water column. So the luminescence that we studied of the benthic animals, the luminescence of these structures that stand up high in the water column, the zoanthids and the corals, is green shifted compared to pelagic bioluminescence. So Sanka managed to get a picture that shows this difference in color. This is the green luminescence from the Gerardia, that zoanthid colonial anemone. This is the luminescence from a pelagic animal that is running into that. So this is green shifted and this is blue. So it's possible that a lot of the bioluminescence that's seen by benthic animals may be that of pelagic animals that are stimulated to glow when they strike these benthic structures. Now getting back to the two crabs with the two visual pigments. These are the two species, Gastropticus and Humanita, and frequently we see them associated with these tree-like structures down at the bottom. I'm going to, fiddle with the lights again. These are three Gastropticus sitting on the sea whip, and these are three Humanita picta sitting on a Gerardia with one Gastropticus right here. So the species that have the two visual pigment systems, at least at the lithoherms, are hanging out on these tree-like structures that the pelagic organisms are constantly running into. And what we also noticed on our subdives is this kind of behavior. This is the little crab sitting right here. And we see this under full lights as well. But what you're going to see it doing is it appears to be picking something out of the water column and bringing it to its mouth to feed. So they appear to be sitting in these branches, not feeding continuously, but only periodically. So the question is, are these crabs that hang out in this greenish benthic luminescence using their two visual pigment system to distinguish between benthic green-shifted bioluminescence, which they obviously don't eat, versus pelagic blue-shifted bioluminescence, which they do appear to be eating and either picking off the structure or picking out of the water column. To determine whether or not this is a possibility, it's going to take some mathematical modeling. And so my colleague, Sanka Johnson, who's a math nerd, is going to do that modeling. And so maybe next year we'll have an answer as to whether or not they can distinguish between yucky green benthic bioluminescence and delicious blue pelagic bioluminescence. And lastly, I often like to take undergraduates out on my cruises, but I frequently don't have any room because ship time is so hard to come by. So Dennis Hanasak, Ned Smith, and I had been talking for years about trying to run an oceanographic experience for undergraduates course. And in 2009, we managed to teach the first course. And this was designed to provide undergraduate students with their first ocean-going experiences. This oceanographic experience for undergraduates is part of our Semester by the Sea program, where FAU students spend a semester at Harbor Branch taking a full load of fieldwork intensive classes for college credit. So the funding for this class was provided by FAU, a grant from the Florida Institute of Oceanography for the ship time, and a very generous donation from Lon and Audrey Kite to teach the class. So because we had 14 students, we divided them up into two groups. And Florida Institute of Oceanography provided funding for a cruise on the RV Weatherbird. This is the first group that went out. This is the second group, dressed in their foul weather gear to work on the deck. Now, the idea is to give them a taste of the trials, the tribulations, and the joy of discovery on an oceanographic research experience. Unfortunately, this cruise was heavy on the trials and tribulations, not quite as heavy on the discoveries. We had howling winds for 2 weeks before the cruise, and howling winds and white water in the inlet. So we made an attempt to get out on the first leg and chugged out, pounded for 2 hours. Finally, the captain called me up and said, you know, we have white water breaking over the fantail and we're not even underway. The fantail is very close to the surface, and he said it's just going to be too dangerous for students out there. So we decided to turn around, and instead of it being a real oceanographic experience, it was an inlet experience. But at least this is what happens on oceanographic cruises. You go with the flow. If you can't do what you plan to do, then you revise your plan and do something else. In the inlet, we could still do our studies. We were planning on going over the shelf and off the shelf, looking at the diversity of animals over the shelf and off the shelf, and compare those with the physical parameters in the water. Ned is a physical oceanographer. I'm a biological oceanographer. It was kind of a nice meld of talents. Instead, we had to do our studies in the inlet so they could do their studies with water flowing into the inlet, water flowing out of the inlet, still study the physical parameters of the water column, still use the instrumentation even though we couldn't go very deep. So these are the students making some meteorological measurements under Ned's guidance. They could still do that. They could still measure cloud cover. They could still measure how cloud cover related to the penetration of light through the water column, how that related to the penetration of light with water coming in versus water going out. We used a noose-ton net, and a noose-ton net is designed to fish at the surface. It's designed to collect the animals that live right at that air-water interface. So the students were responsible for collecting the sample, filtering the sample, bringing it back to the lab for later analysis, and of course, photographing any cool animals that might have come up. We also used a Cape Town dredge, and the Cape Town dredge remarkably showed us that sea urchins were a lot more abundant in the night trawls than the day trawls. So we could still do our day versus night trawls, and they could still do a nice little comparison if that's what they wanted to compare. So they fixed their samples, they brought them back to the lab, they analyzed their samples, and they were taught how to take photometric measurements using a Secchi disc, which pretty much everybody can do, but in addition also using radiometric measurements using a live core. And in lecture, they were taught the difference between photometric measurements, not so good, and radiometric measurements, very good. But to continue on with the trials and tribulations, they also discovered how difficult it is to deploy over-the-side equipment when you're in a rip-snorting current. So in spite of this, we had an excellent group of students, and they managed to come up with 6 different projects. They were taught how to make a scientific presentation quality poster, and those posters are up on the walls right outside as you walk out. And then we had them do a scientific poster session like they would do if they ever went to a scientific meeting. We had the MOA high school students, the Marine Oceanographic Academy students, come in, and our students presented our posters to them. And let me tell you, those MOA students know a lot. They're a tough crowd. So this year, we're going to run our second oceanographic experience for undergraduate course, and I believe this is one of the only ones in the country that actually offers this. Thanks again to support from FAU, another grant from the Institute of Oceanography, and again, another very generous donation by Lon and Audrey Kite. So this work was funded by the NOAA Office of Ocean Exploration, the National Marine Fisheries Service, Florida Atlantic University, Lon and Audrey Kite, and with incredible support from the captain and crews of the Fisheries Service vessel Bigelow, the Seward Johnson, and the Sea Link submersible. And with that, this is a magnificent picture that Brian Cousins took on the last cruise at sunset with the sub coming up. So I'm going to end it here, and I'll take any questions.